structure this talk this morning is it's just going to be pretty brief. I'm going to go into a little bit of theory stuff, but it's not going to be too long. And once I've kind of introduced the topics that we're going to cover, you can go through the worksheet, which has some background stuff and some commands that you're going to run using pretty standard approaches that most bioinformaticians use to analyze data sets. So we're going to be focusing here on basically using high-throughput sequencing in general, taking things all the way from raw reads through variants. And we actually won't be dealing with raw reads in the data set itself. Those are clean just to save time, but think about it as if you're getting your raw data off of kind of an Illumina machine or a sequencing core is distributing to you or something like that. So Mike spoke a little bit about this yesterday with respect to calling variants, but high-throughput data analysis today is primarily concerned about placing reads within a genomic context and then getting likelihoods of variants, or maybe we're interested in structural variation or indels or something, while accounting for various levels of uncertainty in the genome. And error can be introduced at a lot of different stages. So kind of spoke a little bit about sequencing and library preparation yesterday. You know, from kind of first principles and RAD protocol stuff, things that Mike and Paul went over previously, and maybe you've done in some of your own data set generation, that you can inflate your duplication rate. That can have informatic and biological interpretation consequences of your data. How you do your sequencing, whether or not you're dealing with a random distribution of fragments across the genome, or you're subsetting the particular fragments. Maybe you're doing RAD sequencing, and you have, if you think back to kind of the older RAD stuff, high dropout rates, whatever, as Mike said, that might actually mean. So you might have missing data, and how we accommodate that data, how we incorporate that data into our analysis. It's something we have to really think about. But say you've actually got the raw data, and so you've done a really solid experiment. You've got your genomic DNA. You've been able to process it and generate libraries however you want. One of the first things you might want to actually do is take that data, those raw sequencing reads, and clean them in some fashion. And I'll go over a little bit about read cleaning, but this basically involves taking those reads, kind of throwing out some crappy reads, turning them off crappy bases, making sure that the data that we're ultimately throwing into our analysis is going to be rock solid. If you remember from all the calculations we were throwing up yesterday, that the likelihoods of particular genotypes are going to be really dependent upon the reads and the read depth of those positions. So we might expect that making sure that you have the absolute best set of reads possible is going to really impact your analysis to make sure everything's better. Now, if you don't have a genome, or you don't have anything that can be treated as a reference in a general sense, you might want to do a little bit of assembly to get some sort of reference genome. So de novo assembly of particular regions that might be of interest, targeted regions. Things like using transcriptomes, if that was what was used to generate your target sequence, like Jeff just spoke about a little bit. You can assemble those transcriptomes and then go back and map to those transcriptomes called variants. And the reason I bring this up is because when we think about RAD sequencing and how bioinformaticians analyze things nowadays, the most sophisticated genomic analyses are afforded by things with established reference genomes. So I think of it, I kind of look top down. So the stuff I do is going to be across a wide range of model systems. There's going to be a lot of analyses that we might be interested in doing. And then you have these kind of reduced representation approaches, including things like RADs, where we can end up with SNPs to answer questions of interest. But that's going to be a little bit less powerful than, say, investigating really fine-scale patterns of genetic diversity across the genome in a model system like mice or humans, for example. So the better the reference genome you have, the more information you have about not only the genomic context of things, like insertions, deletions, structural variation, regions of repetitive elements, chromosome structure, and most importantly, annotation. So where are the genes? Where are things that are like genes, UTR regions, regulatory elements, that kind of stuff. And can we identify variation that specifically localizes to those regions? So that's why an assembly is important for some analyses. For lots of things, for population analyses, for estimation of things like effective population size and migration rates, things in really non-model systems that I know a lot of you are really interested in estimating. You don't necessarily need that, but at least it's worth bringing in that given that you have these sorts of tools, things can be really powerful. Once you have an assembly or something that counts as a reference, so maybe you're dealing with stacks and you have stacks of reads that are piled up and you can call consensus sequences. You want to go back and map to those reads. By mapping, this is placing the reads within the genomic little bit about that yesterday. And then you might actually want to go through and process the mapping in a particular way that allows you to make sure that you're dealing with reads, again, not necessarily just the quality of the reads, but that we have high confidence that those reads are placed appropriately in the context of our entire genome. You can then go through and do some processing of that. Indels are particularly tricky, informatically speaking, if you're not calling haplotypes. So you can imagine you have an insertion or deletion in a particular base, but your reference genome, whatever that is, has the opposite. So maybe a reference genome position is an A, you have a deletion of that A in your particular sample that you're working with. And then accounting for the appropriate position of that is a little bit tricky. I think I mentioned that a little bit in the handout. You can also throw out things that just you have no certainty about where they are in the context of the genome, et cetera. Then you can go through to variant calling. And statistical variant calling is a computationally very difficult process. So the way in which we call variants, whether using a likelihood-based approach or using a Bayesian approach to get either maximum likelihood estimates of your genotypes or posterior probabilities of your genotypes, requires that you kind of use specialized programs to go through basically every position where you have coverage for, calculate various likelihoods for alleles and alternative alleles, and then figure out whether or not those are true variants. So I'll kind of give an example of that since I have a little bit of spare time. And then post-processing. So this is the filtering that Gordon and Brett and everybody were talking about. So how you take those reads, how you take those variants and make sure that you have high quality variants in the first place.
Just, hopefully this isn't a long answer, but for your post mapping, when you do the indel realignment, I'm presuming that's with GATK?
Yeah, you can do GATK or other approaches, so there are other informatic ways where you don't necessarily need to go through. The way that some established algorithms deal with indel realignment is you identify where an indel is. You basically do a local reassembly of that particular area. That's if you're calling genotypes per position. If you're calling haplotypes using something like haplotype caller, again in the GATK or in Freebase or other approaches, you actually get simultaneous calling of indels and your variants themselves by virtue of collapsing things into haplotypes, so into those extended stretches. And yeah, so just wrap up post-processing, just filtering, making sure you have really vetted sets of variants, and other people speak about that. So what I want to do this morning is introduce a very general workflow for data analysis, but treat it as if you were working in something that doesn't require an established pipeline. I don't like to use pipelines. I found that every analysis is very different. You might want to tweak things at various steps to accommodate certain biases in your data. So by knowing the tools that are used, you can construct whatever type of approach you want to use to analyze that data. A session on this would be lacking if I didn't briefly describe the data structures of some of the common files, SAM files, VCFs, FASTQs. Something I don't think has come up yet. And then perform these basic analyses using the Unix command line. So in the document I emailed you, the commands are in there, and you can copy and paste them in there into your terminal, so you don't necessarily need to sit there and try to get every letter right. But in addition, and I guess this is the fun time to mention this. So in my data folder, you've got the raw reads and a reference genome, that stuff. I also included a folder that's just called test. It actually includes the results of everything at every step. So if somebody has any sorts of problems, maybe something's not working, I'm of course able to help try to troubleshoot things, but you can still look at the files, you can still see what the output would be, and then we could have a discussion about trying to get things pieced apart. And part of my job and what I've done for the past several years as a graduate student and as a postdoc has really been almost consulting about bioinformatics. So I've worked on a lot of different data sets and a lot of different species to answer a variety of various interesting questions and been involved in all sorts of different projects. So if you have questions about things that might be non-standard or how you can analyze your data or how you can get most out of it, please come and ask me. It's what I do. Contact me. Okay, so one thing a lot of people generally want to know is what type of skills should I develop if I'm going to be analyzing libraries and analyzing high-throughput data at a really solid level. So I want to use applications, maybe I need to do a little bit of scripting, write my own. What should I learn kind of from the beginning? So my recommendations, and the recommendations of some of my colleagues and collaborators, is that it's beneficial to learn Unix and learn the command line environment. And this can be any Unix, Linux-based distribution, Ubuntu or Debian, or anything like that. as long as you're comfortable navigating around directory structures, running basic commands, maybe doing a little bit of programming, you're going to be fine. And so coincident with that, you want to know a shell language. And in most cases, unless somebody has a very particular reason why they don't want to, I recommend using Bash. Bash is the born-again shell. It's great for most purposes. Most bioinformatics stuff online is written with people using that shell. I also recommend a scripting language. And my recommendation here is to pick one and get really good at one so that you can do your analysis. Don't try to get just kind of okay at 20 different ones. It's not going to help you. I use R and Python daily. Consider myself intermediate to advanced in those. And Perl used to be really popular back in the day. So that's something people might be useful, or might find useful. I don't like Perl personally, but again, a big one. If you're going to be writing any sort of applications, things where the calculations you're doing are very mathematical, you're going very slow, and you need to really expedite your analysis, I recommend a compiled language like C or C++. In some cases, you might be able to get by with Java. Most of the applications that do a lot of things like de novo assembly, variant calling to some extent, are written in C or C++. Several of them, especially the GATK and Picard and Negro, are written in Java. And this is also something that I've been asked a lot. Where do I go to get help? So Mike was right yesterday. Type into Google. Google's your friend. Figure out how to phrase things appropriately so you get the good answers. But there's going to be three things that pop up all the time. And this is where I spend a lot of time, and I contribute a lot to these communities and help out. So the first is Stack Overflow for general programming questions. This doesn't necessarily relate strictly to bioinformatics, but you'll be able to find lots of resources there that can help you out with your code, help you out if something's going wrong, etc. I contribute very heavily to BioStars. It's kind of a big forum group where lots of people can go in and ask questions about specifically bioinformatic and programming applications. There's a ton of users on there, and people usually get pretty detailed responses. It goes to anything from really basic analysis, what programs can I use to calculate FST, versus I need to do a genome-wide association study, and here's these weird models I'm trying to apply, et cetera, how can I select among things? So it's a really great resource. SeqAnswers has been around a little bit longer. SeqAnswers is slightly different than BioStars in that it also includes stuff about the molecular techniques. So if you're having problems with your protocols and any maybe wet work you're doing in addition to particular informatic work, you can go there and ask questions as well. And of course, it's free to register for all these sites. And none of this is for pay. So, to kind of introduce the exercises, every command I want you to run is going to be in the worksheet, and we're going to be starting from clean reads. Again, I'm going to be going over a little bit how you might expect to do a little bit of data pre-processing. And depending on kind of the flow and how everyone's progressing, we might stop and examine some of the individual data structures. As part of the workshop, I have you looking at the data structures, but as you might expect, there are lots of things in here, tons of information per line. You might have 75 million reads, and there's going to be 75 million lines in a BAM file, lots of additional information, these huge files. So visualizing them can be a little bit tricky, but then also understanding what's actually going on in those files is important in figuring out what's going on with your analysis. Okay, so the data set I gave you is a subset of one of the data sets that I've been working on with Jeff. It's just for one individual, and this is for the Algerian mouse spreads. So we have population level sampling from these. This is in Northern Africa. I picked a single individual. The capture itself was a 55 megabase capture. This is the entire exome. So we didn't capture a subset, we just did the entire exome, and this includes flanking regions as well. So we're interested in regulatory elements like UTRs. The good news for us is that mus spreadus is pretty closely related, just it's not too far related, nothing with the timescales we talked about previously from the mouse reference, So we have a pretty closely related reference. And that means we can really carry over the annotation information from that reference itself, even though we don't have a mus spreadus genome. There's one theoretically coming out from the mouse genome working group pretty soon. But what this means is that we can use these analyses to really hone in on the function and ask questions about where variants are at, what those variants might do, and those sorts of questions. So we have the whole toolkit open to us. I'm terms of being able to answer questions about genomes and do true genomic analysis. And I don't want everybody to deal with the whole 3 gigabase genome and everything, so what I did for this workshop is I took a 20 megabase region of chromosome 1, which is about 50 transcripts that have start codons in this position. So it's going to be pretty small, still bigger than anybody else who might work in some other systems, way bigger than viruses and that sort of stuff. But it should be illustrated with some of the examples. So the way that I generate this data set, just so you might know if you're interested, is I took this entire sequencing effort, and I mapped it to chromosome 1 and the whole genome, and then I extracted every read that mapped to that region, and then threw in some junk reads, so that we have stuff that won't map and things that will map, so it's not like an idealized data set. There's going to be a little bit of this. Okay, and I mentioned that this data set itself, I don't want to spend time doing read cleaning and everything, so the reads in there are actually already clean reads. And I think cleaning reads is incredibly important. So I had a collaborator just a couple weeks ago that was going through and calling variants and was noticing that variants were really clustered around one another. And what had ended up happening is that a graduate student had neglected to clean the reads, and so a bunch of junk reads were going in, and there was variation that was being identified by virtue of either sequencing error or mapping error, any of these sorts of kind of sources of error introduction in general, that were then actually showing up as variants in the raw data, in the final data set. I don't know if it actually has filtered. But there are a variety of approaches to do this, and I talk about it briefly in the introduction to the worksheet. One of my collaborators, Matt Settles, who's at University of California, Davis, has pioneered a pretty good cleaning pipeline. I think it's pretty rock solid and it's fundamentally sound. But basically the way that it works is that you go through and first identify low quality base calls from all of your reads, and you can actually just chop those off. So by low quality base calls, I mean for every single position in your reads, you might know that you also get quality scores associated with it, and those quality scores give us some measure of uncertainty about how competent, or some measure of competence, I guess I should say, about how the machine thinks that that particular base is called correctly. So we can go through and remove anything that we lack competence in. You might also know that you want to remove residual adapter sequences. This is a big one. So if you have residual adapters, if your insert size for your library is small, and you sequence through to your adapters, you're going to have the sequence at the ends of your reads, potentially both, but generally here, where a bunch of reads have this exact same sequence, and then that might actually be picked up as variants. So you can imagine that all this maps to the reference genome, all this is called variants, and that's bad news. One thing I really like to do, informatically speaking, is to take reads and merge reads that overlap. So if a read has, if you have, say you're doing paired end sequencing, you read it have a short insert size, and your first pair goes this, or your first mate goes this way, and your second mate goes this way. Those can actually be combined into a single read. And what that means is that now we're reducing 2 records, our FASTQ file and our BAM file, everything to 1. And then we're still accommodating this information. So we're reducing our file size, we're reducing the number of lines, we're reducing the computational time, and we're making it easier for the programs that we're using to get us those results. And so finally, you can kind of end up in with a good series of sequences. Either you've got few paired end sequences, or you can basically treat these that are overlapped as single end sequences. Maybe you have paired end sequences, one mate fails, you can still keep the other one and use that as viable information. Okay, so I have a little bit here about data structures. I'm not going to go into it a whole lot. And actually, I'll just leave it up on the board, and as people progress through the worksheet, we can stop and kind of talk about these briefly. But what I want you to do now is just take some time, start reading through the worksheet, going through performing the analysis. And I want to mention that with the way that the Docker containers were set up this year, and the way that some people might be performing analyses in their local computers, I also included a little section on how to call programs. So I noticed that there's a little bit of confusion yesterday about, if I have GATK, how do I run GATK command line? And it's not as simple as calling GATK in some cases, but I enumerated those. So if you are running it locally or on the Docker container, depending how things are set up, there might be little differences in the way that certain programs are called. Almost certainly not unless you're kind of going off script, but this should help you not only in this kind of workshop session, but also when you get back to your particular university, your system administrators have things like that. So I'm gonna be here, I'm gonna be walking around and looking to see if people have any help or anything, so, or need any help, so go ahead and start to work through the worksheet, and we have the next